Hi everyone, welcome back. So hopefully everybody survived our winter, Wonderland week well. I'm, really glad to see all of y'all again. So for today, we're going to be talking about our next step in how DNA codes for our life. And so that step is called protein synthesis. So I hope that y'all understand DNA and DNA replication really well, because now we need to talk about how DNA actually codes for proteins. So this lesson is going to be a little bit lengthy just because there are a couple of processes with this. So if you do need to pause the video, come back to it later, just to make sure you understand, definitely go for it. If you need to rewatch the video again, or if you need to set up tutoring with myself or your teacher in order to make sure you fully understand, go for it. Okay, once you get this process, it's actually really easy. So let's go ahead and get started. So the first thing that I'm going to do is I'm actually going to go ahead and show a video just so you can see what's happening in our body. Our bodies are doing this right now because we always have to make proteins. So let's go ahead and watch this video just so that we can check out, what our body is actually doing, inside of our cells.
Stated Clearly presents, what is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind, there's millions of different kinds of proteins. They each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine, AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life. This entire process, as complicated, as sophisticated, as magical as it might seem, is entirely based in chemistry. It can be studied, it can be understood. I'm John Perry, and that's DNA stated clearly.
All right, so hopefully that kind of made sense to you. So what we're going to do is now we're going to go ahead and hop on over to our notebook so that way we can go ahead and fill out our notes that go along with this. All right, so I'm on slide number 14. So we've already filled out the DNA notes, the DNA replication, and so now we need to start on a topic called protein synthesis. So if you recall from that video, if we're talking about protein synthesis, okay, so protein synthesis is the process of creating proteins based off the DNA code. Okay? So we know from our central dogma that DNA makes RNA. Okay, RNA is going to code for our proteins. And one of the things that we need to remember is that the thing that makes up proteins are amino acids. So technically that RNA is coding for amino acids, and then those amino acids will, fold and assemble themselves to make the proteins. So when we're talking about RNA, so RNA is called, stands for ribonucleic acid. It's just like DNA, okay, so it's made out of nucleotides. Okay, and so if I go through and I, formulate my nucleotide, I'm going to do the same thing that I did for DNA. So I have right here a phosphate group. Okay, that's my letter B. I have a nitrogenous base, which right here is letter C. And then I have another 5-carbon sugar, and in this case, the sugar's ribose. So it's not deoxyribose like how it was for DNA. This one's ribose. Okay, so that's one of the differences between DNA and RNA. Another difference is that if I look over here on the right, I have several types of RNA. There's only 3 here that you absolutely need to know. If you take AP Bio, you're going to learn that there's actually a lot more than 3, but we're just going to focus on the 3 main ones right here. So if I look at my strand of RNA up here at the top, I can see that it's a single strand. DNA we know is 2-stranded, it's double-stranded. So RNA being single-stranded is one of the differences. So additionally, if we're looking at this RNA, because we have different types, this first one up here, we actually call messenger RNA. Okay, and that's abbreviated as mRNA. Okay, so messenger RNA does exactly what it sounds like. It's going to carry that genetic message from the DNA outside of the nucleus to the ribosome. The ribosome is what's responsible for assembling those amino acids. So we need to somehow get that message from the DNA outside. And so that's what the mRNA does. So the mRNA is going to take that genetic code from the DNA and it's going to transport it out into the ribosome. Okay, so messenger RNA, so carries, should have made my font smaller. Okay, so carries DNA message out of the nucleus to the ribosome. All right, I'm actually going to make that font smaller. Let me see here. So, all right, and I will underline this right here. All right, so let me do the same thing here. Okay. There we go. Technical difficulties. Okay. All right, so that's a messenger RNA. Okay, so it's going to carry that DNA message out to the ribosome. Okay, and we'll talk about how it does that here in a second. Okay, so this little blob right here, and when I draw this, I usually draw it looking like a muffin. Okay, so this little blob right here is actually called ribosomal RNA, okay, abbreviated as rRNA. And you'll notice that whenever I'm writing mRNA, rRNA, the first letter is lowercase and RNA is capitalized. Okay, so just make sure that you're doing that whenever you're typing out or writing the different types of RNA. So my ribosomal RNA is, again, exactly like it sounds. So this creates the ribosome and reads code from mRNA. So that's my ribosomal RNA. Underline that too. Okay, so my last one. Okay, so this is a weird looking thing. Okay, so this is actually called transfer RNA. Okay, we abbreviate that tRNA. Okay, so tRNA supplies the ribosome with the correct amino acids to create the protein. Okay, that's what tRNA does. All right. So, and you can see right here, okay, so this one's pretty easy to remember because it does kind of look like a T. And so what I have down here at the bottom, you can see I have some bases here, and these bases are called an anticodon. I'll talk about that here in a second. So up at the top, there's my amino acid. So it's going to transfer one amino acid at a time based on that message from the mRNA. So these are three main types of RNA that you need to know. Okay, so remember, for RNA, it's made up of nucleotides just like DNA is. The only difference is that the 5-carbon sugar is ribose. Okay. Also, if we're talking about our types of RNA, we have multiple types of RNA. There's only one type of DNA. And not only that, my messenger RNA, which is the one that looks most similar to DNA, is single-stranded versus double-stranded. Okay. All right, so let's go ahead and move on. So how do our cells actually make these proteins? Okay, so we have two processes for this. We have transcription, because we need to copy that DNA into the mRNA, and then we have translation, where that mRNA is what's giving the message for those amino acids. So transcription, translation. Okay, I think transcription comes first before translation because transcription has a letter C here and C comes before L. Okay, that's usually how I remember it. So with transcription, okay, this one occurs in the nucleus. And remember, DNA cannot leave the nucleus, so we have to have this happen in the nucleus. And so just like how we had DNA polymerase matching complementary bases during replication, we have RNA polymerase this time. Okay, so it matches complementary nucleotides to create mRNA, from DNA. It's early, goodness. All right, and then after that, my mRNA leaves the nucleus to the ribosome. Okay, so one of the things that I want to point out, okay, when I'm talking about RNA polymerase matching the nucleotides, so just like how we had A matches with T, G matches with C, it's going to do the same thing, except that in RNA, we don't have T, we don't have thymine. So anytime we have an A in the DNA, it's going to go to a base called U, which is uracil. Okay, so I'm actually going to put this right here. And I'm going to put a little space. Okay, so G matches with C. I'm going to put that first because that does not change. Okay, so if you have G on the DNA, you're going to make C on the RNA. If you have C on the DNA, you're going to make G on the RNA. Okay, so right here, okay, so T is going to go to A. So if you have T on the DNA, you'll have A on the RNA. But if you have A on the DNA, you're going to get U, which is for uracil. on the RNA. Okay, so remember, RNA does not have thymine, it has uracil. Okay, all right, so that's transcription. We'll practice that a little bit later on. Okay, so now we have our mRNA, it's left the nucleus, it's gone to the ribosome. So how do we code for our proteins? So that process is called translation. Okay, so my mRNA is decoded on the ribosome. And so what ends up happening, my little tRNAs, my transfer RNAs, are bringing the corresponding amino acids. Okay, so how do the tRNAs know what amino acids to bring? Okay, so basically, if we're looking at our mRNA, we're going to group our mRNA into sections of 3. And those sections of 3 are called codons. Okay, so tRNA brings matching amino acids that pair with codons, okay, sets of 3 nucleotides on the mRNA. Okay, and again, we'll talk about codons a little bit more as we keep on going. Okay, so lastly, okay, and then my ribosome assembles the protein. Okay, I told you there's a lot of steps, but once you get this, you're going to get it. Okay, all right, and I have 2 little pictures here to kind of help you understand the processes and what's going on. Okay, so I can see in transcription, which is my first step, my RNA polymerase is going to zip along my DNA and it's going to generate my mRNA. mRNA is going to leave the nucleus and go to the ribosome. I can see here I'm at the ribosome, which is this orange thing. My mRNA is traveling through and my tRNA, you could see there's actually sets of 3 right here. Okay, those are my codons. So it's bringing an amino acid to match with the codon on this mRNA. Okay. All right, so let's look at another picture and go ahead and label. All right, so I have this diagram here, okay, and I can see that I have DNA up here, my double helix, and I can assume this dotted line is a nucleus because that's where DNA is at. And I have this beige looking thing, which I'm going to assume is the ribosome because that's what we're talking about. So I'm going to go through, I'm going to drag and drop, so I know that's my DNA. Okay, so for this process, we have transcription occurring. And so transcription is going to generate my mRNA, which is this one right here, number 2. Okay, so this little single strand is my mRNA. It's going to leave the nucleus, and it's going to go to the ribosome. So remember, I said those sets of 3. Okay, so those sets of 3 are called codons. Okay, there's a codon right there. Okay. So now, okay, my mRNA is in my ribosome. Okay, so right here, I don't know why they drew it like this, but you know, this is what's bringing my amino acids, these little gray things, and I can see the little dark gray balls are the amino acids. So I can assume these little gray things are my tRNA. Okay, so right there, tRNA. So on my tRNA, okay, I have something that matches with these codons. So my tRNA actually possesses something called an anticodon. Okay, and I can't see the little bubble because of my camera. I'm assuming it's somewhere right there. Okay, so this beige looking thing I know is my ribosome. Okay. And my little dark balls are my amino acids. I'm going to move this around. There we go. These are my amino acids. And then lastly, my amino acids will be assembled into a protein. Okay, and then that's going to go and it's going to make us, if you know you're looking at the video, it was going to make that baboon butt. You know, it makes just about everything that's a protein. All right, so yeah, this is one diagram, okay, to help you kind of figure out what goes where. All right, so here's another diagram. This one's a little bit more detailed, and this one is just showing translation. So let's go ahead and look at this. So I know this green thing is my ribosome, okay, and my ribosome can be divided into 2 pieces. And so, it's pretty obvious. I have my large subunit and my small subunit. Y'all don't have to know that. You just need to know the green things, the ribosome. All right. So, again, they drew my tRNAs really weird. They look like little aliens. Okay, so these little blue things are my tRNAs. And I know that because I can see these little balls right here. And I'm assuming those little balls are amino acids simply because over here I see this little thing that looks like beads on a string, and I can assume that's my protein. All right, I have this beige looking strand here that has some different colors as it goes through. And so this entire thing is my mRNA. Okay, and they're kind of confusing with how they labeled this. So I'm going to put mRNA right here. So remember, a set of 3 nucleotides is called a codon. Okay, and then my tRNA has an anticodon. Right? So hopefully starting to sink in a little bit. All right. So go ahead and take a couple of minutes and pause the video and answer these questions. All right. All right. How'd you do? So I went ahead and answered these questions. Okay, so what is the role of mRNA in the process of protein synthesis? We know it carries the DNA message to the ribosome. What is the role of tRNA in this process? It brings amino acids that match the codon on the mRNA. How does the ribosome know the sequence of amino acids? So mRNA has codons, which are sets of 3 bases that code for different amino acids. We're going to look at how to do that here in a second. What happens if you have a sequence that's changed? You could get a different amino acid, and that could, form a different protein. So we'll talk about that one a little bit later. Where does transcription take place? It's in the nucleus. Translation is in the cytoplasm. What's the difference between a codon and an anticodon? So codon is on the mRNA and anticodon is on the tRNA. Okay? So next, summarize the relationship between proteins and genes. Well, we know genes are sections of DNA. So our genes are going to code for mRNA. mRNA is going to code for proteins. Okay? So hopefully you did okay on this. So next, we need to figure out how to decode mRNA. Okay, so this is actually part of the of this unit that's the most fun. Most of the time, students enjoy this lesson. So we need to figure out, okay, how do we do this? So when we're decoding mRNA, mRNA nucleotides are read in triplets called codons. We've already gone over that. Okay, so scientists use a codon chart. I don't actually know why I made this into 2 blanks. Okay, that was my bad. Okay, scientists use a codon chart to read the sequence of mRNA. Okay, so how I use this chart, okay, you can see that I have my 4 bases. So remember U, because we don't have T, so I have uracil, cytosine, adenine, and guanine. I have that on the left side, the top, and on the right side. I also made a note that right here on the left, that's where my first base in the codon is going to be. My second base on the codon is on the top, and my third base in the codon is on the right. Okay? All right, so whenever I'm trying to create a protein, I always have to have a start point. And so I know, again, it's just from me being a teacher for so long, okay, my start codon is AUG. Okay, so my AUG also codes for an amino acid, and that's when my protein knows when to start coding. So if I look at AUG, okay, so what I'm going to do, okay, so here's my first base. Okay, this is my first base in the codon. I'm going to look for the letter A, okay, because my codon is AUG. So there is my letter A. My second base in the codon is U. Okay, so now I'm going to come up here at the top and I'm going to look for a U. Okay, you can see it's right here, this first column. So now I match up that A and U, and I can see I'm in this box somewhere. Okay, so I have ILE, 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 MAT. Okay, so those all stand for different amino acids. Okay, so ILE is isoleucine, MAT is methionine. Again, I know that because I've been a biology teacher for a long time. So my last letter in my codon Okay, remember my start codon was AUG, is G. So I'm going to come over here to the right-hand side. I'm going to look at this third base, and I know it's the letter G. So I'm going to trace that G back to the box I was in, and I can see I have MET, which is methionine. Okay, so AUG is my start codon. That's why I went ahead and highlighted this. I also have 3 stop codons. Okay, so if I have a start, I have to have a stop. So I can see that my stops are bolded right here. Okay, so if I need to figure out what those codons are, okay, so again, okay, I see I have the red stop boxes up on this top row. So I'm going to come to my first base. First base is U. Second base, okay, I can see right here I have an A. Okay, third base, if I come over here to the right, if I trace it back, there's another A. Okay, so I know that UAA is 1 stop codon. I have 2 more. Okay, so again, U, A, and then this bottom one, which is G. Okay, so UAG is another stop codon. And then my last one, okay, so U, come up here, G, and then A. Okay, so UGA is my 3rd stop codon. All right. Okay, let's do some more practice because I know this is weird. This is crazy. Okay. All right, so I have an mRNA sequence right here. So I have AUG, AGG, GUA, AUA, UAA. Okay, so perfect thing is that we know this is mRNA because there's the letters U there. Okay, if it had the letter T, I would say that's DNA, and we need to convert that to mRNA. All right, so I have my notebook from last year that has my codon chart in it. Okay, so that way I don't have to flip back and forth. Okay, so you may have to flip back and forth, but I have a codon chart. Okay. All right, so first off, okay, I have AUG. If I go back over here, I know AUG was my start codon, which I highlighted in yellow, which is MET, methionine. Okay, so I'm just going to put that right here, MET. Okay, methionine. Next, I have AGG. Okay, so AGG, okay, if I go right here, first base, my first base is A. Second base is G. Third base is G. Come back here, I can see it's ARG, okay, arginine. Okay, so right here, arginine, ARG. All right, try to figure out what GUA is. Did you get it? Okay, it's valine. Okay. AUA is isoleucine. I actually don't know if that's the correct abbreviation. Yes, it is. All right, and then my last one, UAA, is a stop codon. Okay. All right, so we're going to be practicing this a lot, okay? So if you don't get it, if you need some help, don't worry, we're here to help you, all right? So if you need to rewatch this video, watch some of your Amoeba Sister stuff, I have a bunch of little videos here that will potentially help you, and I hope you all have a great day. Bye.